tubulin. We have centrioles. One centriole keep on elongating, and that is Dyenin. Rows of Dyenin proteins. This is a complete spermatozoa. Doom. So in the previous lecture we had discussed how mature spermatozoa forms, and the tail or flagellum of that sperm, it forms from the organelle called centriole. In this video we will discuss in a bit more detail that how flagellum forms and how flagellum works, right? And we will start it with the discussion that how centriole, what is the structure of centriole, right? But before that, let me tell you an interesting thing. The only cell of human body that contains flagellum is sperm. We do not have any other cell that contains sperm. That contains flagellum, right? So this is about flagellum in human, but in other animals, in small animals, especially in bacteria, different cells, there are lots of flagellum. They contain lots of flagellum, right? So let's discuss how centriole works. So here is the structure of centriole, and centriole consists of these tubules, these tubes, and this each tube, right, this tubular structure, this each tube is called microtubule, and we have 3 microtubules forming a triplet, right? So triplet structure, 3 microtubules that form a triplet, and we have 9 such triplets in one centriole. 1 2 3 4 5 6 7 8 9, you can count these. We have total 9 triplets, right, that forms this centriole, and this is the structure of one centriole. Similar to that, we have another centriole that lies perpendicular to this, 90 degree to this, and both these centrioles, they are called centrosome. So a centrosome consists of 2 centrioles that are arranged perpendicular to each other, right? And if we look at a structure of a triplet, it consists of 3 microtubules, and each microtubule, we will discuss structure of single microtubule, it is made up of these small bead like proteins. You can see these globular proteins, right? It is made up of these globular proteins, and these are called tubulin proteins, right? We have these tubulin proteins consist of 2 subunits, alpha subunit and beta subunit. So this is alpha, let's say alpha tubulin, and this is beta tubulin, right? So these alpha and beta tubulin, they are arranged in a particular fashion to form microtubule, right? And this microtubules, these 3 microtubules arranged to form a triplet, and we have 9 such triplets, and they are arranged to form one centriole and another centriole that is present perpendicular to this, right? Now let's see how from this centriole flagellum arises. For this we will go to our beloved software Blender 3D. So here we have, okay, so this is a cross section of, what is this, spermatozoa, right? And it consists of, it is actually developing spermatozoa right now. So it consists of, what is this thing, acrosome, and behind the acrosome, as I have told you, that this is nucleus is present, right? And here we have centrioles, one centriole and the second centriole, and each of the centriole, they consist of what, 9 triplets, 1 2 3 4 5 6 7 8 9, interconnected with each other, right? This is a structure of centriole. Now from this centriole, flagellum will arise. How flagellum will arise? These microtubules, they can be elongated, they can further enlarge if we add more tubulin proteins here. And how tubulin proteins are added here? Very simple. Just like any other tubulin, any other protein of the body, tubulin proteins are also synthesized by ribosome. You know, they're from DNA, mRNA is formed, and from mRNA, as we have discussed previously, from mRNA, ribosome transcribe them into mature protein or tubulin proteins, and then these tubulin proteins, they can come here and attach here, right, like this, to elongate any of these microtubules, right? They can elongate any of these microtubules. Now out of these triplets, only two are elongated, right? Like this, only two are elongated. Let's see how. You see, out of these three triplets, 3 microtubules forming triplets, 1 2 3, only 2 elongates, 1 2, right? Here also 1 2, right? So now we have in the flagellum, we have not triplets, not 9 triplets, but we have 9 doublets. We have 9 pairs of microtubules, and we do not just have pairs, but we have 2 central singlets, 2 central single microtubules. Don't worry, I will discuss that what is their function very soon. Don't worry that what they are doing here. I will just discuss that, right? So they keep on elongating, and in the center we have singlets as well, and they keep on elongating until a complete tail is formed. But before that, let me discuss another thing. We do not just have these tubulin proteins here. There are many other proteins that are associated with flagellum. I will not discuss each and every protein that is associated with flagellum, because that way the lecture will become very long. I will discuss here the most important one, and that is Dyenin. Let's hide the mitochondria. You know, mitochondrial sheath is also present here, right, surrounding this spirally arranged mitochondria here, right? And we have two rows of what is this, Dyenin proteins. We have 2 rows of Dyenin proteins, so they are also called Dyenin arms. They have arm like structure. So this is outer layer of Dyenin, this one, this is outer layer of Dyenin, right, or outer row of Dyenin, and this is inner row of Dyenin arm. So these 2 rows of Dyenin, they are arranged along the long axis of these doublets, these pairs of microtubules, right? So this is the structure of flagellum, the cross section, right? 9 plus 2, 9 doublets and 2 singlets, along with Dyenin arms and some other proteins that keep them together, right? And finally, let's complete this, that it will form a complete tail, right? And now we have, what is this? This is a complete spermatozoa. So what is the structure of complete spermatozoa? This is acrosome, this is nucleus, this is centriole, and this is flagellum, and spirally arranged mitochondrial sheath that surrounds the base. And this tail, like this inner mechanical part of flagellum, these microtubules along with associated protein, it is as a whole, it is called axoneme. Now axoneme, along with this, surrounded by this membrane, is called flagellum or tail, right? And this is the structure of flagellum, and this is the complete structure, complete 3-dimensional structure of spermatozoa, right? Now let's discuss how it works. Now we will discuss that how this flagellum works, right? And for this I will use my fingers to demonstrate this. Now come here. So these are my fingers, of course you know this, and this finger, let's suppose this is 1 microtubule, right, and this is second microtubule. So 2 microtubules form the doublet of flagellum, and here also these 2 microtubules, they form doublet of, what is this? Doublet of flagellum. So we have 9 such doublets, but we are discussing here about 2 neighboring doublets, what they do to each other, these nasty neighbors, right? So this doublet, it is the one that is lying in front of this. So this is front doublet and this is back doublet, right? And here these are Dyenin arm of this doublet. I have not shown Dyenin arm here, but these are the Dyenin arm of this doublet. And with the help of these Dyenin arms, these doublets, they can crawl on or climb on the neighboring doublet, right? Clear? So let's suppose that this is the rest position, right? This is the rest position, and in the rest position, the mid doublet, mid Dyenin, this Dyenin, in the rest position, this mid Dyenin, right, it is lying in the front of this, it is lying at the dot position. It is lying at the dot position of this previous one, the doublet that is behind it, right? So this is the rest position. And now when it crawls or climbs on this doublet, the previous one, and it will what will happen? It will reach above. From dot it will reach, this middle one, it will reach to star position. So from dot position it will reach the star position, right? But while doing so, it has risen upward. It rises upward, right? But fortunately this does not happen. Why it does not happen? Because all of these doublets, they are anchored into the base. All these doublets, they are anchored into what? Into the centriole, into the basal body, and they cannot move up or down relative to each other, right? This cannot rise up or this cannot go down. Clear? So this middle Dyenin, it still wants to touch this beautiful star. So how it can touch the star without rising upward? Normally you should rise upward to touch the star, right? But it cannot rise up. So when it moves, when it crawls on this, when it crawls on this, it can still do this, but while doing so, because it cannot rise up, it bends like this. It bends forward. It bends forward like this. Right? Now it has touched the star, but it has caused a local bend forward. And in this way, a local bend backward can be generated if this crawls on forward one. Right? So due to this local bends, what will happen? The flagellum will move forward and backward. And you know, we also have two central singlets. What is the role of these central singlets? It is not clearly known, but it is said that it controls this mechanism, right? It tells the doublets that which doublets should move, which doublets should rest, and which doublets should crawl. So due to the controlling mechanism of those singlets, some of the doublets, they remain resting while other crawl on them, and vice versa, right? So this is the mechanism of flagellum, movement of flagellum. So you have seen in that analogy, in the fingers analogy, that what happens is that these, let's suppose these 3 or 4 doublets, these front one, these 3 or 4 doublets, at one time they are resting, and these previous, those that are present behind these 3 or 4 doublets, they are moving, they are crawling on those forward ones, on the forward lying doublets. And in this way, due to this crawling of this previous one on the forward one, this causes backward bending, and vice versa, that if front ones are crawling on the back one, then there is forward bending of flagellum. So, and what control this mechanism? These central two singlets, right? So in this way, forward bending and backward bending. So forward and backward bending lead to a wave of movement, right? It causes a wave of movement in the flagellum that leads to motion of, that leads to the particular specific motility of flagellum of sperm. So this was about how sperm moves, right? Now finally I would like to discuss another point, that as we have discussed previously, that all the cells, as all the sperm precursor cell or spermatozoa precursor cells, they undergo incomplete cytokinesis. I have told you in previous lecture that they undergo incomplete cytokinesis, meaning that they are connected by cytoplasmic bridges. So here also these sperm, they are also still connected by cytoplasmic bridges, right? You can see here. And finally, when they completely mature, then what happens, that they separate from each other, right? So sperm, they are still embedded into the Sertoli cells, but they are now separate from each other, right? And finally, when they are mature, what happens, that they come out of these Sertoli cells, these furrows of Sertoli cells, and they are released into lumen of, what is this? Lumen of seminiferous tubules, right? So what happened here, they were embedded into these furrows, and then they come out of these furrows and release into lumen of seminiferous tubules, and this process is called spermiation. What is spermiation? Spermiation simply means release of spermatozoa into the lumen of seminiferous tubules, right? Then what happens, that, so finally, when they are released into the lumen of seminiferous tubules, then from all these seminiferous tubules, the spermatozoa, they ultimately come into epididymis, right? They come into epididymis, and then from epididymis through vas deferens, they are released with ejaculate into they are released outside the body through the urethra, right? So that's how spermatozoa forms. Now I have a little question here, that I have told you that in seminiferous tubules, the sperms, they are protected from the hazard of immune system with the help of blood testis barrier. Now what thing protects the sperm in epididymis from immune system? You got my question, that in seminiferous tubule there is blood testis barrier. What is in epididymis that prevents it from immune system? So I will address this question in next lecture. Meanwhile, you have to tell me the answer to this question in the comment section. So this was about spermiogenesis. Finally, another point that I would like to highlight here is that the role of testosterone. I have told you that testosterone has very important role in maturation of sperm. Testosterone stimulates the Sertoli cells. It causes the Sertoli cells to release certain growth factors that causes maturation of sperm. But it has direct role in spermiogenesis as well, right? From the process of transformation of spermatid to spermatozoa is called spermiogenesis, and it directly causes spermiogenesis. They have found certain receptors here in the spermatids. They have found certain receptors, and these are the receptors of, what is this? Testosterone. So that is why scientists do believe that it has a direct role, testosterone has a direct role in spermiogenesis as well. Thank you so much for watching this video. If you like this video, please press the thumbs up button. Please give your comments regarding this video, and please subscribe to my channel. Thank you so much.